from Stanford University. Professor Chaudhuri obtained his bachelor's degree from Berkeley in engineering physics, and then he went on to obtain a PhD degree, again at Berkeley, also in collaboration with the University of California, San Francisco, in bioengineering. And his postdoctoral fellowship was at Harvard University in biomaterials. And actually, this sequence really nicely explains, nicely aligns with the evolution of his research interests. He was recruited by Stanford in 2013 as assistant professor at the Department of Mechanical Engineering, and was promoted to an associate professor at this department, and also appointed as associate professor in the Department of Bioengineering, and a member of the Cardiovascular Institute at Stanford, which is really famous after the quality of their research and their conferences. Professor Chaudhuri's research is at the intersection of mechanics and biology. His lab is interested in the molecular mechanisms that give rise to the complex mechanical properties of cells, extracellular matrix, and tissues. They're investigating how complex mechanical cues can influence cell division, differentiation, and the progression of cancer. To this end, they exert and also measure and control forces that are acting on materials and on the cells at the nanoscale, and they develop biomaterials for three-dimensional cell culture that allow a precise manipulation of mechanical properties. Professor Chaudhuri has an excellent publication and citation record with over 19,000 citations and an excellent H index of 43. When you look more closely into his published work, you will see that his most impactful work are the 3 papers that he published in Nature and an additional 6 papers that he published in Nature Materials. These papers collectively report the development of new biomaterials, regulation of cell and tissue behaviors by mechanical forces, either directly or through the extracellular matrix. And each of them was cited as many as 400 to 2,100 times. This line of research continues in recent years, again with publication in Nature Family Journal, and you can see a shift towards studies of cancer and fibrosis, individually and in combination. Not surprisingly, Professor Chaudhuri received a number of honors and awards, and I will just mention a few. He obtained Career Award from National Science Foundation. He obtained Rising Star Award from BMES, Biomedical Engineering Society, Merit Award from the National Cancer Institute. This is really a big one. He was elected to be Hellman Faculty Scholar. He was elected to speak at the Engineering Symposium of the National Academy of Engineering, Frontiers. He obtained Young Faculty Award from DARPA and National Research Service Award from the NIH. Thank you so much for being with us today, Professor Chaudhuri, and you can now take over the screen and start your presentation.
Screen. Well, thank you so much for that extensive introduction, Gordana, and also the invitation to be a part of this really wonderful seminar series. I very much enjoyed listening to some, excellent talks from some of my favorite scientists over here over the last few years since I found out about this Tissue Talk series. Okay, so just wanted to jump in and start with a broad motivation of some of the questions that my group is interested in. Of course, as many of you on participating in this and listening to this seminar series will know, over the last several decades, it's emerged that it's not just what's inside the cell that's important for cell biology, but what's outside the cell, the microenvironment, has emerged as a really critical regulator of cell fate and function. Now, some features of that microenvironment we're particularly interested in is dimensionality. So there have been some couple seminal studies shown here, but many other studies have shown that a three-dimensional microenvironment is critical for capturing the physiologically relevant behavior, at least in some biological context. And mechanics also plays a role. So, in particular, matrix stiffness. So matrix stiffness has been found to play a critical role in regulating differentiation, cancer progression, neuronal branching, proliferation, apoptosis, and various other processes. And this has been shown in hundreds, if not thousands, of other papers at this point, and has been shown using 2D culture, 3D culture, animal studies, and many of the predictions have been borne out in analysis of human patients. Now, this has been, of course, I think had a tremendous impact and been really important, especially in the bioengineering community. However, we know that tissues aren't purely elastic materials that can be fully defined by an elastic modulus. They exhibit complex mechanical properties, including viscoelasticity, which is what we've been focused on. So viscoelastic materials, as the name suggests, exhibit some responses characteristic of elastic solids and also viscous liquids. And in particular, they exhibit a time-dependent mechanical response. So that's a key property of viscoelastic materials. Some examples of viscoelastic materials are jello or gelatin and silly putty. As you see, there's a time-dependent mechanical response in this silly putty ball over here. And so just some evidence to support the premise that tissues and matrices are viscoelastic are shown in this panel here, where we plotted the loss modulus, which is a measure of viscosity, versus the storage modulus, which is a measure of elasticity for various reconstituted extracellular matrices, soft tissues, and skeletal tissues as well. And for all of these materials, we see that the loss modulus at 1 Hertz is about 10% of the storage modulus at 1 Hertz as well. So all of these materials exhibit a substantial viscoelastic quality or response, or they're all viscoelastic. So these measurements are all done at 1 Hertz. And you see kind of a similar loss tangent or ratio from the loss modulus to the storage modulus. But at longer times, you see some variation in the viscoelastic responses of these different tissues. So this becomes evident in stress relaxation tests. So in a stress relaxation test, we apply a constant deformation to a material, and we measure the stress over time. So in this measurement, the stress corresponds to the resistance to deformation. So we see is that in an elastic hydrogel, the stress stays constant. So the resistance to deformation is time independent. Whereas for these various soft tissues, we see the stress is relaxed over time with a characteristic time scale ranging from seconds to thousands of seconds. And so again, this corresponds to the resistance to deformation relaxing over that time scale. Now, there's various ways to describe viscoelasticity, and we typically refer to describe viscoelasticity in terms of stress relaxation. And the reason is because it gives us somewhat of an intuitive idea of how cells might respond to viscoelasticity. So if we imagine a cell first binding to a substrate and pushing or pulling on that substrate, initially, there's some resistance as defined by the initial elastic modulus or stiffness. But over time, that resistance to deformation relaxes as related to these stress relaxation properties in the material. Now, mechanistically, there is many origins of viscoelasticity. Basically, any mechanism that dissipates energy gives you a viscoelastic response. So some of these mechanisms include unbinding of weak non-covalent bonds in the matrix, release of polymer entanglements, or protein unfolding. So the recognition that tissues and reconstituted extracellular matrices are viscoelastic has motivated the two questions that my group has been interested in since we started at Stanford back in 2013. And these questions are, how does extracellular matrix viscoelasticity impact cells? And we're in particular interested in the visco side of viscoelasticity, since we already know a lot about how stiffness impacts cells. And we're particularly interested in contexts in which we think a three-dimensional microenvironment might be important. And then we're also interested in the physics of cell matrix interactions during dynamic processes, such as cell migration, division, as well as morphogenesis. So today I'll talk about 4 stories. So the first story is more of just a series of stories. So I'll present a few snapshots of some of our published work looking at the impact of matrix viscoelasticity on cells. A lot of this published, so I'll go through this fairly quickly. And then I'll go into three more recent stories, a couple published and some more recent unpublished work. Okay, so just to jump into it, I'll first talk about our work looking at the impact of matrix viscoelasticity on cells. Now, this topic is something I became first interested in as a postdoc in David Mooney's lab at Harvard. And there I work closely with Luo Gu, who now has his own group at Johns Hopkins. And to address this question of how viscoelasticity impacts its cells, we first needed to develop a material system in which we could independently tune that stress relaxation or loss tangent independent of the initial elastic modulus or stiffness. So the materials approach we came up with was based on alginate hydrogels. So alginate is a polymer derived from algae. We can cross-link it into a nanoporous hydrogel using divalent cations such as calcium. Now, importantly, these are ionic cross-links. They're very weak. They can unbind and rebind quite easily, making these hydrogels intrinsically more quite viscoelastic. Now, some other important features of alginate are that it's inert. So cells have no way of binding to alginate itself, and they can't degrade alginate, or at least mammalian cells can't degrade alginate using proteases. However, we can couple cell adhesion peptide motifs such as RGD to allow cells to bind to the alginate through integrins, or we can form interpenetrating networks with things like reconstituted basement membrane matrix or type 1 collagen to form a bioactive matrix. Okay, so the idea we came up with was that if we altered the molecular architecture of these alginate gels, and in particular, varied the molecular weight while varying the ionic cross-linking density, we hypothesized that we could independently tune the viscoelastic properties. And indeed, that's what we found we could do. So if we look at these stress relaxation curves on the top panel here, we see that as we go from high molecular weight alginate to low molecular weight alginate, we get faster stress relaxation. So we can quantify this by looking at the time at which it takes for the stress to relax to half its original value. And that varies from thousands of seconds down to about a minute. That overlaps with the physiological range that we saw earlier. We can also look at the loss tangent of these matrices, and that varies from about 0.12, 0.13 in these high molecular, or sorry, 0.06, 0.05 to 0.06 in these high molecular weight gels, to about 0.12 to 0.13 in these low molecular weight or low molecular weight PEG gels. So again, it overlaps with the range that we see physiologically. Now, importantly, we can vary these properties independent of the initial elastic modulus, or that initial resistance a cell feels as it first pushes or pulls on these hydrogels. So that's shown here. So the modulus in these gels is around 10 kilopascals. We can, use other formulations and vary the modulus from hundreds of pascals to about 20 kilopascals, while having a range of viscoelasticity that we can access. So importantly, these hydrogels are relatively stable over 1 to 2 weeks. And if we do fluorescent diffusion studies, we see that the diffusion release kinetics are somewhat similar between these different gel formulations, suggesting at least a similar pore structure. So this isn't the only way to vary viscoelasticity in these hydrogels. We've developed a few other approaches, and we've published a protocols paper. So hopefully it will be useful for anyone who's looking to use these same approaches. Okay, so as I mentioned, we spent a lot of time, and we've published a few papers looking at the impact of viscoelasticity on cells, and just kind of showing some snapshots from these different studies here. And we started out by looking at differentiation of mesenchymal stem cells, and we see that if you have a stiffness of 17 kilopascals, and you go from slow relaxing to fast relaxing hydrogels, we see that the osteogenic differentiation, as indicated by staining for alkaline phosphatase, is enhanced. And we also saw that the osteogenically differentiated cells were also able to form a more bone-like matrix consisting of type 1 collagen and mineralized matrix, so something that looks a lot more like bone. Similarly, when we culture chondrocytes or cartilage cells in these hydrogels, we see that again in the fast-relaxing hydrogels, the cells are able to secrete a lot more type 2 collagen and aggrecan here in yellow, a much wider volume of interconnected cartilage-like matrix, essentially. And so from these two areas that we've studied, we can appreciate how viscoelasticity might be a potent design parameter for biomaterials we use in regenerative medicine. We also see an impact of viscoelasticity on also cell spreading and adhesions, matrix remodeling, mitosis, cell cycle progression, as well as cell migration. And from this impact on, and as well as on apoptosis and morphogenesis. Now, from the impact we see of viscoelasticity on cell division and cell migration, we might expect that changes in viscoelasticity could play a role in cancer progression, since cancer progression is intimately tied to proliferation and growth as well as invasion. And indeed, that's what we found. So this is a study led by our collaborator, Natalie Turok, who's here at Stanford. And what we found was that so for patients with non-alcoholic steatohepatitis and type 2 diabetes, what you see is that those tissues have the same stiffness relative to normal healthy liver, but enhanced viscoelasticity. And what Natalie and Jerome, the first author, found was that enhanced viscoelasticity activates malignant signaling and increased proliferation and invasiveness in those liver cells. Okay, so this is all published, so I wanted to spend my time, and some of these papers are quite dated, so I wanted to talk about more recent work. But before I do that, I just want to take one slide to talk a little bit about what we've learned about how cells sense matrix viscoelasticity. So, one mechanism is we find that cells bind to matrices through integrins, apply contractile forces on the matrix through actomyosin and Rho-mediated contractility, and through that mechanism sense both stiffness and viscoelasticity in 3D. So this is similar to what has been observed in 2D culture as well. And so those pathways are still active in 3D and in the context of sensing viscoelasticity. We also see that changes in cell volume are involved. And so these are studies led by Hongpyo Lee, as well as Sungmin Nam, who's a former PhD student who's now a professor at Michigan. And what we see is that in 3D, cells will change their volume. And as their volume expands, that activates mechanosensitive ion channels, which allow influx of calcium ions, which activate downstream signaling pathways to change the cell phenotype. And importantly, when you have a purely elastic matrix that's confining, we see that cells are often stuck and they can't change their volume. Whereas when the matrix is viscoelastic or degradable, cells are able to change their volume and activate these pathways, and in that manner are able to sense their confinement and matrix viscoelasticity. Some other highlights of what we found is that mechanotransduction does occur independent of YAP in some contexts. So that's what Joanna Lee, a former postdoc in my group, found. She's now has her group at Genentech. And we've also found a role of the epigenome in mediating mechanotransduction. So these are studies led by Ryan Stowers, a former postdoc in my group who's now at UC Santa Barbara and has his own group. And he found that when in response to changes in matrix stiffness, in this case, there are broad changes in chromatin accessibility, which functionally mediated mechanotransduction and mediated a change in phenotype. Okay, so those are all studies that are published. So I wanted, and somewhat older, so I wanted to go with some more recent work. And the recent story I'll talk about is work led by Dharaj Indana, a former PhD student in my group who's now a postdoc at Caltech. And Dharaj, when he joined my group, was interested in development and was following this exciting development of the use of pluripotent stem cells in these increasingly sophisticated models of development, particularly in early stages of embryonic development, where we see that human development deviates quite substantially from mouse development. And so in this context, pluripotent stem cell-based models of early embryogenesis provided a powerful approach to understanding some mechanistic insights of these early stages. So what Dharaj was interested in was whether we can use engineered hydrogels to develop better models of certain stages of this early embryonic development. And if we are able to develop these models, can we generate some new mechanistic insights into early embryogenesis? Okay, so what we first did was cultured human-induced pluripotent stem cells in hydrogels with the range of viscoelasticity, stiffness, RGD ligand density. And we immediately see from bright field images that after 7 days, we see some striking differences in the morphology of these iPSC clusters that form. And we can put together a phase diagram where we see that in slower relaxing matrices with lower ligand densities, the cells primarily undergo apoptosis, where at higher ligand densities and with faster relaxation, the clusters are able to form a lumen, as shown in this nice immunohistochemical image on the top right here. Okay, so we published this story and became interested in these lumen-containing structures because they started to look a lot like what we see in the early human epiblast. So the epiblast is a structure that occurs early in development from day 7 to day 11. We see it emerge. And it consists of pluripotent stem cells that initially start out as a ball and end up eventually forming a lumen. Now, and some characteristics of this early human epiblast are that it's a single layer of pluripotent stem cells. There's a lumen, and these cells exhibit apicobasal polarity. And all of these features are what we see in our in vitro model, where we see the cells maintain their pluripotency. They have the single layer, and they exhibit apicobasal polarization. We also see some similarities in the nuclear architecture. So the size of the nucleus and some features of the nucleus are similar between our in vitro model and in vivo, and closer than what we find from culturing these pluripotent stem cells in matrigel, for example. And we also see the biologically relevant scaling between the number of cells and lumen volume and kind of how rapidly these clusters expand. So for all of these reasons, we think that this epiblast-like structure that's forming in these hydrogels may serve as a nice model of the early human epiblast. And so under that idea, we said we wanted to use this model to try to gain insights into what causes some of the biophysical mechanisms involved in early embryogenesis, early epiblast development, including what drives early lumen expansion in these structures. We can see a video of this lumen first forming in bright field in this video on the right. Okay, so lumens form across development, and so there's been a lot of study on the origins of lumen nucleation and expansion. And it's thought that really the primary driver of lumen expansion across development are osmotic pressure gradients. So under this idea, once you have a nascent lumen, cells can pump in osmolytes into the lumen that increases osmotic pressure, forming a gradient with the outside, which draws water into the lumen, expanding the lumen. Now, what's required for this mechanism to occur is that you need to have sealed lumens, so that the junctions between the cells need to be tight. So we first tested whether we were observing tight junctions in our epiblast-like structures. And so we added fluorescent dextran to the media in our hydrogels and wanted to see whether that entered the lumen readily or was it excluded if there were tight junctions. What we see is that the dextran, fluorescently labeled dextran, enters the lumens quite readily when the structures are small. So for smaller lumens, it appears that the junctions are not tight, they're actually leaky. And indeed, we can confirm that using bleaching studies. We bleached the fluorescently labeled dextran, and within minutes, fluorescent dextran from the media enters into the lumen. So what this tells us is that we don't have tight junctions in these smaller epiblast-like structures, so that osmotic pressure gradients cannot drive lumen expansion. And this is not the only piece of evidence that we have some other evidence to support this idea as well. So if it's not osmotic pressure gradients, what could drive this early lumen expansion in these epiblast-like structures? Now, one clue was that associated with this lumen expansion, so here we see this lumen expanding in these clusters, what you see is that the overall cluster is expanding as well, so it's pushing out on the hydrogel. And these are fairly stiff hydrogels on the order of 20 kilopascals. So this lumen expansion mechanism must be associated with some kind of a force generating mechanism. So that led us to start to look at different force generating mechanisms and whether they could be associated with luminogenesis. So we first started out by looking at actomyosin contractility. So we inhibited actomyosin contractility using various inhibitors and found that it had no effect on luminogenesis, as you can see from these quantifications here. And so actomyosin contractility is not driving lumen expansion in our model. So we then asked, what about actin polymerization? So we know actin polymerization can generate force, especially when it's in the form of polymerization of actin networks, such as dendritic actin networks or bundled actin networks. So we first looked at the structure of actin in our epiblast-like model. And so using light chain microscopy, we visualize actin and we see this dense actin meshwork that's forming at the apical surface, so on the lumen. And we see microvilli poking out from the cells into the luminal space. So we then inhibited actin polymerization using inhibitors of the ARP2/3 complex and NWAS, which are mediating formation of a growing dendritic actin network, as well as formins, which are involved in growth of a kind of more bundled actin network. And what we see is that inhibition of any of these components potently reduces and inhibits the formation of clusters with the lumen, suggesting the role of actin polymerization in luminogenesis. Okay, so and this was really the only class of inhibitors that did inhibit luminogenesis. So that was promising, but then we then thought about how could actin polymerization be driving opening up of a lumen? And so we thought that maybe what's happening is that if you have actin polymerizing at this apical surface, and it's polymerizing laterally, then cells are essentially pushing laterally on one another, and maybe that opens up the luminal space. And so kind of maybe a macroscopic analogy to that is this video of this group of people in a circle, and they're pushing on each other, and the space in between them is opening up. So similarly, the cells are pushing on one another. The apical actin growth is providing this lateral force that's helping to open up this luminal space. So I will note this is a video we got from YouTube. I did not make my students perform this demonstration. So the hypothesis is that maybe actin polymerization at this apical surface pushes on a given cell pushes on the neighboring cells, opening up this luminal space. So to assess whether this was the case, we first, or Daraj first did some painstaking time-lapse imaging, looking at the actin dynamics. So here is the actin in black and the bright field video and over on the left-hand side. Let's play that again. Okay, and so what we find is that often lumen opening is associated with apical actin growth of individual cells. So here we see the apical surface growing on an individual cell, and that's opening up the lumen here. So it is lumen opening expansion can be driven by the behavior of single cells and is not necessarily a global, driven by the global activity, coordinated activity of cells. Okay, so that's suggestive that consistent with this possibility of this actin growth mechanism. But to get more insights, we conducted some laser ablation studies. So we ablated the apical actin meshwork in an individual cell and looked at what happened afterwards. So here we see a video here. The network's ablated, and we looked at the dynamics of the cluster over time following ablation. So to interpret this video is fairly complex. So instead, we put together a chymograph where we're looking at the actin signal in the ablated cell, as well as the neighboring cells. So at the apical surface. And what you see is that immediately following ablation, in the neighboring cells, they tend to grow into the ablated cell. So those neighboring cells expand until the actin network recovers in the ablated cell, and then the ablated cell pushes back and recovers its kind of the apical dimensions it had previously. So these dynamics are indicative of this lateral pushing behavior between the cells, which we hypothesized could drive a lumen expansion. Okay, but to further assess whether this was reasonable, we collaborated with Vivek Shenoy and his postdoc, Andrei Zakharov, and they put together a fairly comprehensive model of these pluripotent stem cells and these epiblast-like structures. And they conducted some simulations, and the two key results they had were that one, actin polymerization was sufficient to drive lumen expansion, and two, a key mediator of this working is actually the rigidity of the actin meshwork. So you actually need a fairly relatively stiff actin meshwork for that apical and lateral actin growth to prop open this luminal space in the center. Okay, and so our overall picture is that actin polymerization in the early embryo drives a lumen opening, or at least in our epiblast-like model. So of course, it's hard to do any studies in vivo, but there are some in vivo sequencing data that indicate that we have an increase in expression of actin polymerization related genes at the stage of epiblast morphogenesis, where we expect this mechanism to occur. So it's not definitive evidence, but at least it's suggestive that this may be the case. Okay, and there's our video again. And so let's move on to the next story, which is work done by Julie Chang, a former PhD student in my group who's now at Genentech, and Ashrith Saraswati Patla, a current postdoc in my group who will soon be starting his own group at University of Minnesota. And their work has been looking at the initial invasion of the basement membrane during breast cancer progression. So during breast cancer progression, this initial invasion of the basement membrane represents a really critical step. So the basement membrane is this thin layer of matrix consisting of laminin and type 4 collagen, and it separates epithelial tissues from surrounding collagen-rich stromal matrix. Now, this represents a critical stage because if you have a pre-invasive carcinoma, breast carcinoma, it's very treatable, and most of the patients survive. Whereas if you have patients who are found with invasive breast carcinoma, there is a substantial mortality rate in those patients. So this is really important biologically. And it brings up a really interesting physical question. So cells are on the order of 10 microns in size, whereas the pore size of these basement membranes can be on the order on the nanometer scale. So, tens to hundreds of nanometers. So how is it that cells invade and migrate through these pores that are 100 to 1,000 times smaller than the cells themselves? So an analogy I like to use here is this bunny trying to make its way through its playpen, and we can see it squeeze through this very small pore, but of course there's a limit to how small that pore can be. And similarly with cells, there's a limit to how small those pores can be, where they can just squeeze through the pores. Okay, so we're not the only ones to think about this question. There's been a lot of work in this area, and kind of the current thinking is that to invade through basement membranes, cells use a combination of proteases, which biochemically degrade the matrix, so essentially dissolve the matrix, and mechanical forces to kind of mechanically open up space in the basement membrane to squeeze through. However, there are some limitations in these studies, and really the major focus of these previous studies have been on single cell invasion of the basement membrane. Whereas what we know is that initial invasion of the basement membrane during breast cancer is largely collective in nature, at least in many cases. And so these are just some histology slides provided to us from our collaborator at Stanford, Rob West. And what you see is this is the invasive front of invasive breast carcinoma, and you're looking at staining for E-cadherin, so cell-cell junctions, and you see there's a very strong staining. So these are cells are maintaining their connections to other cells as they initially invade. Okay, so the question we had was, what are the role of proteases and forces in initial invasion of the basement membrane by collective groups of cells? So to address this question, we developed a 3D culture model of collective invasion of the basement membrane. So we took MCF10A cells, which are a model of a mammary epithelium. We cultured them in reconstituted basement membrane matrix or matrigel, and they form these organotypic acinar structures. So the cells form a lumen, they exhibit apicobasal polarity, and importantly, they form their own basement membrane. So that you can see on the bottom panels here, where we stain for laminin 332 and type 4 collagen, two key components of the basement membrane. And we see the layering with laminin on the inside, collagen 4 on the outside that we expect of basement membrane. And importantly, the thickness of the laminin that we see in our in vitro model matches what we see in pre-invasive breast cancer or ductal carcinoma in situ, but it's a little bit thinner than what we see in normal tissues. But of course, we're trying to capture this invasive breast carcinoma condition. So we then take these organotypic acinar structures with a basement membrane, and we cultured them in hydrogels that were either soft or stiff. The soft hydrogels, the clusters stay rounded. In the stiff hydrogels, we see that the cells invade collectively through the basement membrane. So in blue, what we're seeing in this video here is laminin 332, and in red, we're seeing the actin cytoskeleton. And you can see that the cells are invading and puncturing through the basement membrane and invading into the surrounding matrix. Okay, so then now that we had this 3D culture model, we sought to understand what are the mechanisms driving this collective invasion of the basement membrane. So we first looked at proteases. So we inhibited protease activity using broad spectrum protease inhibitors. And we see there's an reduction of invasion, but invasion wasn't completely abrogated, suggesting that proteases are involved, but they're not strictly required for invasion to occur. And indeed, if we looked at the signal of the basement membrane around the invasion site, we see that it's higher and densified, consistent with the idea of cells using forces to open up the basement membrane, in which we expect the matrix and basement membrane to be densified around that site of invasion. Okay, so if forces are involved in basement membrane invasion, where could those forces be coming from? That was the next question we had. So what we noticed is that the cells were a lot larger in these invasive structures relative to the non-invasive structures, so about 2 times larger. And so we thought, could that be involved? So we inhibited cell volume expansion by inhibiting mechanosensitive ion channels, which are involved in cell volume regulation, such as TRPV4, or increasing osmotic pressure to prevent that volume expansion. And in both cases, when we prevented volume expansion, the structure became less invasive. Conversely, if we induced volume expansion in non-invasive conditions, that promoted invasion, as you see here. So using hypotonic media, invasion is enhanced. Okay, and so you might ask, well, this is just a 3D culture model. Do we know this is relevant in vivo? And of course, that's always a challenge with in vitro models, and there's a limit to what we can do to test the relevance. But what we did was we analyzed cell volumes and using cell cross-sectional area as a proxy for volume in normal tissue ductal carcinoma in situ, or pre-invasive breast carcinoma, and invasive breast carcinoma. What you see is as you go from normal to pre-invasive DCIS, you get an increase in cell area similar to what we see when we go from the soft to stiff IPNs. So we do see an increase in volume, area and volume during breast cancer progression. But interestingly, what you'll notice is that between pre-invasive breast carcinoma and invasive breast carcinoma, the cell volume or area is similar. This suggests that the cell volume increase is necessary, but maybe not sufficient to drive invasion, and maybe there's other mechanisms involved. So along these lines, we looked at the role of actomyosin-based contractility coupled to the basement membrane through integrins. So we blocked integrin binding to laminin using these blocking antibodies that reduced invasion. And similarly, if we inhibited Rho-mediated actomyosin contractility, that also reduced invasion. And then finally, when we used FRET-based sensors of Rho-A activity, we see right around the invasion site enhanced Rho-A activity initially during the initial kind of opening up of the basement membrane. And so all of these together suggest the role of contractile forces due to actomyosin contractility on invasion. But this brings up a really interesting question. So we talked about how expansion is driving invasion, but now I'm arguing that contractility is also driving invasion. And these seem to be 2 kind of competing phenomena. So how is it that both expansion and contractility could be working together to drive invasion? So we struggled with this question, and then we came up with the thought that maybe it's due to the directionality of these forces. So we did some imaging of the basement membrane and looked at basement membrane deformation and kind of deformations that are occurring in the radial versus tangential direction. And what we found is that the basement membrane was deforming to a much greater extent in the tangential direction, so in the plane of the basement membrane. And so what that suggested to us is this following model. So as cells expand their volume, that's stretching the basement membrane that's surrounding these structures. And then cells are exerting local actomyosin contractile forces to pull on the basement membrane locally, largely within the plane of the basement membrane to further stretch it and potentially then rupture the basement membrane. And so that's kind of our working model of how those 2 volume expansion and actomyosin contractility work together to drive basement membrane rupture and collective invasion of the basement membrane. Okay, so for the final section of my talk, we'll talk about some of our work looking at single cell migration in confining viscoelastic matrices. So there's been a lot of interest in confined migration by single cells, so cells migrating in environments where which are confining. So that means that the largest dimension that they have to migrate through is smaller than the cell itself. And what we've learned from many of these studies is that there's really a rich set of behaviors that cells can adopt and display when they're migrating in these confining contexts, and something you don't see in 2D culture or kind of unconfined migration. However, and while we've learned a lot from these studies, most of these studies use these geometries where where you have a pre-existing, predefined path for the cell to follow. So, for example, these studies use these microfabricated channels, which have a dimension of microns for the cells to then, in terms of the diameter of that channel. And so the question we've been interested in is, what about when cells have to migrate in a confining microenvironment where there's not a pre-existing path available? So in other words, when there's there's not a micron scale opening, there's instead a nanoporous surrounding meshwork. And so we know this question becomes relevant when the pore size or channel size falls below around 1 to 3 microns. And of course, we know that cells in these contexts can use proteases to biochemically degrade the matrix. But what we've been interested in is, what about the role of matrix mechanics and kind of, and in particular, we started with the hypothesis that maybe mechanical plasticity of extracellular matrices could allow cells to migrate through. So matrix mechanical plasticity is the ability, refers to the ability of a matrix to be mechanically remodeled or be malleable. And kind of our idea was that maybe when matrices exhibit mechanical plasticity, cells can simply push and pull and mechanically open up a channel to then migrate through. Okay, so before we test the hypothesis, first I wanted to present a slide demonstrating that the idea of mechanical plasticity is relevant to extracellular matrices and tissues. So we can measure mechanical plasticity by using creep recovery tests. So essentially, we apply a mechanical stress to a material, we hold it, and then we let it go. And we look at if there's any irreversible or plastic deformations remaining over long times. And what we've shown is that reconstituted extracellular matrices in various tissues all exhibit some degree of mechanical plasticity. Okay, so our first test of this hypothesis occurred a few years back. So this is work done by one of our first PhD students, Katrina Wisdom, who's now at GSK. And she looked at the migration of breast cancer cells in nanoporous hydrogels, so alginate-based hydrogels, with tunable plasticity. And so here's just some videos of these breast cancer cells. And in green, we're looking at the actin cytoskeleton. And what you see is in the low plasticity matrices, cells stay rounded. They don't really do a whole lot. Whereas in the high plasticity matrices, some substantial fraction of cells are able to migrate quite robustly through the matrix. And she did some extensive mechanistic studies. We see that cells are forming these invadopodial protrusions that push and pull on the matrix and eventually open up these channels that they are then able to migrate through. So this supports this idea, and that if a matrix exhibits a sufficient amount of mechanical plasticity, then cells can simply use forces to open up channels to then migrate through, independent of proteases. Okay, so we then have more recently been exploring this question in the context of immune cells, because we know that immune cells are thought to rely less on proteases to migrate. And so the first study I'll talk about is work done by Kolade Adebowale, a former PhD student in my group who's now a postdoc at Harvard, but soon to be starting his own position at UCSD, and Cole Allen, a current PhD student in my group. And they've been looking at the migration of monocytes. So monocytes are immune cells that circulate and then traffic into our tissues where they can differentiate into macrophages or dendritic cells to mediate immune responses. So we cultured monocytes in interpenetrating networks of type 1 collagen and alginate. So the idea is that the alginate gives us a handle with which to tune the mechanical properties, whereas the collagen, the collagen gives us this fibrillar collagen network that is relevant to the stromal matrices that these monocytes will migrate through in our tissues. So we see in these videos that these monocytes we can see some of these cells are migrating quite robustly. We can track the migration, and we see that both increase in stiffness and faster relaxation enhances the migration of these monocytes. So we've done some mechanistic studies to try to understand the mechanisms mediating this monocyte migration. So what we see is that the monocytes migrate independent of adhesion. So if we remove the collagen and we look at migration in alginate alone, the speed is essentially the same. So they don't need integrin binding ligands to migrate, which is something that's very common and typically observed in immune cells. If we inhibit actin polymerization and actin-myosin contractility, that inhibits migration. If we look at the actin cytoskeleton dynamics, what we see is that there's dense puncta of actin at the very rear of the migrating monocytes. And if we do some stainings, we see that opposite of that dense actin puncta, we see WASP. And then finally, if we do some traction strain microscopy experiments, so looking at how the matrix is deformed around these migrating monocytes, what we see is that ahead of the leading edge of these monocytes, there's evidence that the monocytes are pushing in front of the leading edge. And so kind of overall, what these studies suggest is that as these monocytes are migrating, we have WASP at the leading edge that's initiating formation of this growing dendritic actin network. And this generates protrusive forces that's pushing on the hydrogel. And then the myosin activity is acting to induce retrograde flow of this actin so that it accumulates as this dense puncta at the rear. And then in terms of the force balance, we see that cells are pushing off their rear in many cases, but sometimes off of their sides. Okay, so I'll end this. So I'm running short on time, so I just have one last a few more slides to go through. And so if you notice, when I've talked about cell migration, I mentioned a few different properties. So stiffness, viscoelasticity, and plasticity. And we can visualize all those properties very nicely together in a stress strain curve. So here we're looking at the stress versus strain for a hydrogel. We see initially there's a linear relationship between the two. So this is the linear viscoelastic range where we can get a storage or elastic modulus as well as a loss or viscous modulus. At a certain point, the material starts to undergo plastic deformation, so at a yield strength. And we enter a regime known as the plastic regime, where the material undergoes plastic deformation. And finally, at some stress, which in a shear test would be the shear strength, the material fails, material undergoes failure. So what Byung-Hung Ha, who's a current postdoc in my group, has been interested in is which of these properties is relevant and regulates the migration of T cells in confining nanoporous matrices. Again, matrices where there's no pre-existing migration path. So unfortunately, we don't have the capability of modulating each of these parameters independently. But what Byung-Hung did is he formed a set of hydrogels that had a range of stiffness, viscoelasticity, yield strength, plasticity, as well as shear strength. And importantly, no two parameters are perfectly correlated in this set of formulations. So what that means is that these are distinct properties, as we would anticipate, but also if we conduct migration studies in these range of hydrogels, we can potentially relate the migration behaviors to individual properties, as you'll see. Okay, so Byung-Hung first started by culturing Jurkat cells in these hydrogels. In red, we're looking at the actin cytoskeleton. In blue, we're looking at the nucleus. So we get migration, and we can track the migration speed, the percentage of cells that migrate, the distance they migrate. And he did a number of correlation studies between these various migration characteristics and these various matrix properties. And strikingly, what he sees for each of these migration characteristics, the strongest correlation is with shear strength. And there's actually very little correlation with stiffness and stress relaxation or the loss tangent. And this is these studies shown here were done with jerkats, but we see similar results with primary T cells, although we find primary T cells can generate a lot much higher forces. And kind of our interpretation of these ideas is that cells need to rupture the matrix, or these T cells are rupturing these nanoporous matrices in order to make their way through them. Okay, so I think I'm about out of time. And so I'll maybe skip the summary and just like to acknowledge I did very little of the work I talked about. So done by the fantastic students and postdocs in my group, both current and former, as well as many of our amazing collaborators, both at Stanford, as well as outside of Stanford. And of course, have to acknowledge our funding sources. So I think we do have a few minutes left, and I'd be happy to take any questions.
Yeah, we do. Ovijit, thank you so much. This was really, really wonderful. Let me just quickly introduce our next speaker. So next Wednesday, we will have Eileen Fuchs speaking to us. She is, as most of you know, like a real pioneer in understanding skin from all possible aspects, from cell biology to regeneration. You have a lot of questions, so I will just try to go through a few of them, and then we would like to ask you, like we ask all speakers, to please respond to those that we cannot make offline. We will give you contact information for people.
Okay, will do.
First question comes from Amin Abdul Khani. How does the cell know its own volume? What is the mechanism?
How does the cell know its own volume? That's a very philosophical question. Yeah, it's a good question. I think yeah, it's a good question. I guess I don't have a perfect answer. I mean, I think mechanosensitive ion channels can function to help sense volume changes, so kind of regulate volume changes. So that's one mechanism. I think there's been reported in the literature, kind of cells can be very sensitive to the concentrations of certain components, such as retinoblastoma protein, for example. I think it's when that becomes diluted enough that initiates this trigger to cell division. So, and I think, yeah, so, how cells know their volume, maybe it's, I don't have a good answer. I know that mechanosensitive ion channels, our work indicates that's a mechanism that they can sense relative changes in volume. But kind of absolute volume sensing, yeah, that's a good question. At this moment, I don't have a good answer.
Okay, so question from Dimitris Misirlis. Have you checked in your basement membrane invasion model for the effects of cell-cell adhesion? Are there any tangential forces that you were able to detect?
So we'd say if the question was on cell-cell adhesions in our basement membrane invasion model. Yeah, so if we perturb cell-cell adhesions, we lose that inhibits that invasion process. So invasion is substantially reduced. So it does appear that it's a collective invasion process and that you need cells working together to drive invasion. We haven't, I think what would be cool to do is to try to measure tension between the cells and using maybe FRET-based sensors. I think that Alex Dunn over here at Stanford has developed. I think that would be really cool. I think it's challenging to use those in 3D, but something that we're certainly thinking about doing, and it would be very interesting to explore kind of the more at more of a molecular scale how forces are transmitted in the context of basement membrane invasion.
Okay, Giovanni Datri is interested if you have any recommendation about the most effective test that can be used to measure the viscoelastic properties of the extracellular matrix made by the cells that are cultured inside the hydrogel in 3D.
Yeah, I think that's a very big challenge. So, cells, I think as fantastic work from Jason Burdick, Eileen Gentleman's groups have shown that cells secrete a lot of matrix when they're cultured in 3D, and they're often and then they respond to that matrix that they're secreting. So, kind of what are all the mechanical properties of those matrices? I think it's a challenging question to address in 3D because so bulk measurements obviously won't be effective there. Techniques like AFM, which are, very powerful for measuring, microscale mechanical measurements, you can't access that matrix because the outside of that endogenous matrix is your gel. So I think, it's challenging. I don't really know a good way to make those measurements. I think that's the big challenge for the field.
Okay, so Pierre Nassoy is interested, how come that your epiblast-like structures, these lumen-containing cysts, are most often not spherical, but they're a kind of pointy. They have pointy ends. And he says because the osmotic pressure is the driving force for lumen expansion at these late stages, does it mean that there is also some heterogeneity within the structure?
Yeah, so there's a little bit to unpack there. One thing is quite interesting that we do see these asymmetric structures. They're ellipsoidal, some cases more football shaped. And so, we have some ideas that we're trying to work on to try to understand why it is, but I don't have a definitive answer to say yet of why that happens. But it is something that's, consistently that we observe. So it is quite interesting. And as the question pointed out, we do see a transition to osmotic pressure-driven growth at later stages. So I think what we find is that the tight junctions mature, they seal, and which occurs around a lumen size of around 12 microns in diameter. And at that point, then cells utilize osmotic pressure to expand themselves further. So I didn't get the last part of that question, but yeah.
Okay. You got a question from Mel Vaughn about the role of cell division in the processes that you're measuring, including the lumen size changes.
Yeah, I think it's a great question. So what we see is the lumen expansion occurs fairly rapidly. So in some cases, cells are dividing, some cases cells are not. So it seems to be decoupled at those earlier stages from lumen expansion. But later on, it turns out to be a driving factor for the overall expansion of the cluster and as well as the lumens. So there's a transition to kind of cell division driven lumen growth at later stages.
Okay, and maybe the last question, so we stay within the time and let you go. Asia Guzman, she is asking why the non-cancerous cells, so this is this MCF10A line, invade through the basement membrane. Have you looked into potential changes in the phenotype, like that these cells are gradually acquiring malignant signatures in terms of gene expression and probably other
Yes, I think it's a great question. So MCF10As form these organotypic acerine structures and are typically considered models of normal mammary epithelium. What we and others have found is that we see that they invade when you increase stiffness of the matrix around, and that's what we and others have found to induce this kind of more proliferative and invasive phenotype in the cells when there's this increased stiffness. Not this exact system, but in a very similar system, we've done some, extensive gene expression analysis, and it does seem like, we've done some extensive analysis. You see activation of kind of malignant signaling pathways like PI3K, evidence of, transcriptional activity of factors like STAT3, which are associated with malignancy. And also, if you look at kind of more broadly, we see a set of genes that are upregulated that you see upregulated during in ductal carcinoma in situ. And so kind of this more pre-invasive cancerous condition. So, we do have, so it does appear this increase of stiffness is activating some of these malignant signatures in these cells.
Great, thank you. We still have about a dozen questions that we will be sending to you. So
okay, great. Yeah, happy to answer those. And any other questions that come up.
Yeah, thank you so much. And then, everyone will get an answer. And obviously, please feel free to contact our speaker with any additional questions. Thank you so much, Ovijit, again. And thanks, everyone, for attending and for very insightful questions.
Thank you so much for the opportunity. Really appreciated the time to be here and all of you for sticking on till this point.
Thank you.
Thanks.